The 2021 Wisconsin Science Festival. Hybrid, hands-on, and virtual. Events throughout Wisconsin for all ages. Discover the wonders of science and the power of knowledge with a focus on fungi with scientists, live Q&A sessions, science experiments, and more. Plus, PBS Wisconsin Education launches 2 new interactive labs in the Meet the Lab collection. Full schedule at wisconsinsciencefest.org. Curiosity unleashed.
Hello, fellow scientists. I'd like to welcome you all to the Wisconsin Science Festival field trip, Power from Plants. Thank you so much for joining us today. My name is Ashley from the Wisconsin Science Festival team, and we're here with our special guest, Rebecca from Wisconsin Energy Institute for this field trip experience. So how can you participate today? Well, you'll notice that your picture and audio are muted, but if you have any questions you'd like to ask, you can use the Q&A feature and the chat. As soon as you have a question, go ahead and type it in right away and we'll ask the question, or I'll ask the question for you out loud during the field trip. This field trip will be recorded, and as always, we ask that you do this in all activities at the Wisconsin Science Festival with a responsible adult. We'd also like to thank all our sponsors and partners that help make the Wisconsin Science Festival possible. You can find out more activities and events during the festival at wisconsinsciencefest.org. With that, we're excited to get started. Take it away, Rebecca.
Hi, everyone. I'm Rebecca. I am a scientist at UW-Madison in the Wisconsin Energy Institute. And I am a botanist, which means that I study plants. And in particular, I am interested in plant biology and plant biochemistry, which means that I'm studying plants at a really microscopic scale. I'm looking at individual cells in the plant to determine what they do, how they function, what compounds they're making, and how they regulate the synthesis of those different compounds. My journey to becoming a botanist probably started when I was a kid. I grew up in Winnipeg, Manitoba, Canada, which has a very, very long, cold winter. So when the spring and the summer would come around, we'd be really excited to spend a lot of time in the outdoors and just enjoying nature. But also, my mom is an avid gardener, which means that every single year she would have a huge vegetable garden and really big flower gardens. And she would also do all the planting of flowers in our neighborhood as well. And I loved going with her to greenhouses and nurseries so I could see all the different varieties of plants and how they grew, and just all the different features of the plants really amazed me. I didn't like working in the actual garden as much. There were always a few too bugs for my liking. So I knew I wasn't meant to be an entomologist, but I really enjoyed looking at the plants. My decision to become a botanist really hit home when I was in high school. I was taking a biology class and we did a unit on plants, and something about it just clicked in my brain. I think I was studying for the photosynthesis part of the unit, And it just amazed me when I thought about how plants can take energy from the sun and they can turn it into all the sugars and the food that they need to survive, that they're taking carbon dioxide out of the atmosphere and they're producing oxygen. And as I was sitting there surrounded by my mom's houseplants going, these plants are the reason that I'm alive and able to survive on Earth. And that's amazing. And I thought I just wanted to study more about that and learn more about that. After high school, I followed a pretty straightforward academic path. I got an undergraduate degree, also in botany. And during that time, I spent a little bit of time doing some microscopy analysis, but I wanted to get more experience with research and do a little bit more studying the genes that are in plants and also doing some biochemistry. So I decided to go ahead and go to graduate school, and I got my PhD in botany as well. And then I came to the United States, to UW-Madison, to join the lab that I'm currently in to do the research that I'm in. So I have a presentation here that I'm going to share to tell you a little bit about the research that I'm part of and how we do the research. And if you have questions at any time, as was stated earlier, please put them in the chat or the Q&A. I'm happy to answer questions as we go along. So I work in the Wisconsin Energy Institute, which is sort of on the west side of campus. If you're familiar with campus at all, it's towards the hospital. And we are, for campus, a relatively new building. And this is a building that was designed with clean energy in mind. We house a lot of different researchers that are from a variety of fields of study. So there are plant biologists like myself. There are also a lot of chemists in the building. There are microbiologists and bacteriologists that are studying smaller organisms like bacteria and yeast. We have ecologists who are looking at ecosystems and how plants and the other organisms within the ecosystems interact with each other and how we can grow plants to ensure sustainability. And we also have engineers. Because we are the Energy Institute, we're interested in all forms of renewable energy. So there are scientists who are working on solar power, wind power, nuclear power, and then a big chunk of researchers in the building are working on energy from plants. And I'm one of those researchers. I'm part of the Great Lakes Bioenergy Research Center, or GLBRC. And one of the main questions that we asked for the GLBRC is, can we make fuels and chemicals from plants in such a way that we're going to be removing carbon dioxide from the atmosphere instead of making fuels and chemicals from fossil fuels as we currently do, which is adding carbon dioxide to the atmosphere, which, as many of you may have heard, that is increasing global warming and causing climate change all over the world. So what types of fuels and chemicals are we talking about from plants? What would these be called? Well, these would be biofuels. And our hope would be that these would be at least carbon neutral. We would grow the plants, which are taking in energy from the sun to make all the energy and the sugars that they need in order to grow. We would harvest the plant material. That plant material would be turned into a fuel. Then the fuel could end up in your car or in a plane or in whatever it needs to be in order to keep the world turning. Even better than carbon neutral biofuels would be if we can make them carbon negative. In order to make them carbon negative, we have to be taking more carbon dioxide out of the atmosphere than we are putting back into it. This is something that's achievable with plants because plants have a remarkable ability for carbon storage. They can store carbon as starch, and then they can store carbon in their plant cell walls, which we'll talk about in a few slides. And plants also have these incredible root systems that are equal in biomass, or the amount of plant material, to what you actually see above ground. So a lot of that carbon storage actually happens underneath the soil where you don't even see it. This is why it can be so devastating when whole tracts of forests are taken up and whatnot, because all that carbon storage that was being kept in the plants is removed and ends up being released back into the atmosphere. We have a number of dedicated bioenergy crops that we're interested in to make biofuels from. And there are a couple different features to these plants that we try to hit. So these plants can include sorghum, which is shown out here in a field. You can see a person standing next to the sorghum holding a pole. This sorghum is a type of sorghum called Bioenergy sorghum. It has a lot of plant cell wall material, and these plants can be 6 to 8 feet tall. So they produce a lot of biomass just in a single year. There are also perennial grasses that we're interested in, or mixed prairie, we sometimes call it, switchgrass, and also poplar trees. And these are our main bioenergy crops. The reason that we're interested in these is that a lot of these crops hit at least 2 of the 4 features that are really important for us. One is we want these plants to be native to areas that we're going to grow them. If they're not native, then we're introducing exotic plant species into environments where they don't belong. That can result in species quickly becoming invasive because it can just completely disrupt the ecosystem that they're put into. Secondly, we'd like some of these at least to be perennial. That means that we plant them and then they grow for multiple years before they have to be planted again. This is obviously going to be true of a tree, and switchgrass and mixed prairie also have these features. Sorghum does not. It is an annual plant. However, it grows very quickly. It produces a lot of biomass. So we just know we can get a lot of biomass out of it because it performs photosynthesis very efficiently. The other feature we're interested in is low input. This means that we don't have to provide a lot of fertilizer to these plants in order for them to grow. This would be in direct contrast to corn, for example. In order to grow corn, you have to put a lot of nitrogen and phosphorus into the soil in order to make the plants happy and to have them grow and be productive. With a lot of our bioenergy crops we're interested in, you don't have to use as much fertilizer to still produce productive plants. We're also looking in terms of inputs at the amount of water it takes for this plant to grow. Sorghum is really drought tolerant, and this is great, especially as there are parts of the country that are becoming more and more susceptible to drought as global warming occurs. If we can grow sorghum in those places, then that would be an ideal bioenergy crop there. We're also interested in growing these in such a way that they would be in a polyculture. This means we're growing multiple plants all together, different kinds of plants all together. As soon as you increase the diversity of plants, you increase the diversity of everything else that's in the ecosystem with those plants, with the fungi, the other animals that are around, the microscopic organisms, the bacteria. It's beneficial for the entire ecosystem, and it makes the growth of the plants more sustainable. The way that we're planning to grow these plants, therefore, is not what I'm showing you in this picture here. This is an example of a monoculture. We're seeing sorghum that is growing in a plot at the Kellogg Biological Research Station in Michigan. Where we would actually be growing these plants when we're not doing research on them, if we're growing them for biofuels, is in what we call marginal or non-agricultural lands. So this is land that is either too nutrient depleted in order to grow agricultural crops there, or food crops there, or in lands that are, for other reasons, we can't grow food crops there. So therefore, our hope is that we're not going to be taking away any arable land that could be used for food crops to grow our bioenergy crops. The GLBRC has what we call a pipeline to it, and this starts with our energy crops. And it starts with taking the compounds out of the energy crops that we're interested in. This is the lignocellulosic material, which I will talk about in the next slide. Once we have that material, then we convert it into bioproducts and into biofuels. My role is in this energy crops portion of this, and I'll show you right now what exactly I'm interested in doing. All bioenergy crops, all plants actually, have a plant cell wall that surrounds each cell. This is different than an animal cell. So if you think of an animal cell, it has a plasma membrane around it. A plant cell always has a cell wall that surrounds the plasma membrane. This provides greater rigidity and structure to the cell wall. Ooh, there we go. The cell wall of the plants can vary a little bit, but the ones we're particularly interested in have 3 different components to them. They have cellulose, hemicellulose, and lignin. The cellulose is really the backbone structure to the cell wall. It's made up of individual sugar molecules called glucose that are linked together. Those glucose molecules in a chain then pack together to make what's called a cellulose microfibril. Those microfibrils then stack together to make the backbone of the cell wall. Now, that the cellulose microfibrils alone won't provide very much rigidity. So there is hemicellulose in the cell wall, which is another sugar-based polymer that's in the cell wall. The hemicellulose has some structural similarities to cellulose in that it has a lot of glucose in it, but it has other sugar molecules and it has different bonds and different linkages. And its main function is it surrounds the cellulose and it pulls those cellulose microfibrils together as well, kind of holds them in place. It acts as kind of a weak glue or, just some Velcro to hold the cellulose microfibrils together. The last part of the cell wall is the one that I study, and it is lignin. Lignin is not a sugar. Lignin is part of a different group of compounds, which are called phenolic compounds. It has completely different monomers that are in it. It has a wide variety of linkages that can happen between these different individual units. It's a big, unwieldy polymer that ends up it acts as the glue or the cement that just ensures that cell wall stays together. Lignin is one of the most important components, both to plant evolution on land and also to maintaining really strong cell walls, which allowed plants to remain strong as well. The easiest way that I can think about lignin and the strength of lignin, because it's one of the world's most strong compounds, is think about any wood that you have around you. So you've got a wood desk or a wood chair or just a wood shelf sitting around you somewhere. That wood is composed of hundreds of thousands of millions of cells. And each of those cells has a cell wall. The cells in the wood that you're looking at this point, they're dead. There's nothing left inside them anymore. But that cell wall remains intact. And that's mostly because of the lignin that's there. And the strength of the wood is because the lignin provides a strength to each individual cell wall. So collectively together, that's what makes the wood really, really strong. So for a plant, lignin is absolutely essential. Lignin is what's going to ensure that it can grow to whatever height it needs to, and it can withstand winds blowing it around and rain, torrential rain coming down on it. But from an industrial perspective, trying to get to the sugars in the cell wall, because we use these sugars, the cellulose and the hemicellulose, to make our fuels, the lignin poses a huge problem. We have to get it out of the way so that we can get to the sugars. Ideally, what we want to be able to do is this. This is a cartoon of our cell wall. We want to treat it with enzyme or with some chemicals, and we want everything to break into pieces. Right now, the way the cell wall looks is some of those sugars will break into pieces, the bits that are not attached to the lignin, but the lignin is going to remain relatively intact. What I aim to do is to find ways that we can make it easier to break apart the lignin. And what that involves is finding enzymes that can make compounds that will end up in the lignin, and that will make the lignin easier to break apart. So I'll pause here for a second, and if there are any questions that people want to ask, I can answer those quickly before we head on to the next section, where I talk a little bit about how things work in the lab.
Yeah, so if anyone has any questions, please put them in the chat or in the Q&A. We had a question, but I think it's been retracted, but okay. It was like, how could you make the fuel? But maybe you can just summarize quickly what you went to and how the process of making the fuel, how the fuel is made.
Sure. So I am not one of the scientists that's involved in making the fuel. There's scientists that are on other floors in our building who are doing that. And I would encourage you, if you're interested, to reach out to them because they're always really excited to talk about their research. Basically what they do is they use, depending on the lab, they use either chemicals, a series of chemicals, or they use different enzymes to break apart the sugars into their individual components. And once they have their individual components, a lot of times they use bacteria to eat those sugars. And once the bacteria eat those sugars, they convert it into different fuel types like butanol. Or gamma valeriolactone is another one that we work on a lot here.
That's really cool. So there is like quite a lot of steps to get to that final product, right?
Yeah, yeah. Okay, well, if there are no other questions right now, then I will continue on and take you on a little tour of the lab. Okay, so before I take you on a tour through the lab, if you just think to yourself for a minute, or if you want to type in the chat, what sort of things would you expect to see in a lab that is working on bioenergy or making fuels or growing plants?
Yeah, while people are answering that, we did have another question come up in the chat, Rebecca, and it says,
oh yes, do different plants create different fuel?
Yeah.
Generally speaking, no, because we're using the same sugars from every plant. So we're using the celluloses and the hemicelluloses from every plant to make different, to make the same sort of fuels. Where we can get differences is when we're making what we call bioproducts. So those are other compounds that we can get from the cell wall. And a lot of bioproducts come from the lignin. So it can depend on what the lignin looks like, which is really variable to what bioproducts we make. So the fuels are kind of more general. We're making one type of fuel and it depends what bacteria we feed it to what kind of fuel we make, but we can make a lot of different bioproducts from different plants.
So it's the bacteria that produce the different biofuels?
Sorry, I missed that.
Is it the bacteria that produce the different biofuels then? That's how you get different products in the end.
Yeah, yeah.
Super cool. So we have some answers in the chat to your question. What type of things might you see in a bioenergy lab? If you guys haven't answered already, please place it in the chat, but we do have answers like plants, lights, tools to measure energy, scientific models, all really good answers. Safety equipment.
And then, yeah, you can see as we walk through the lab if you see any of those things. The other thing that I'm always on the lookout for when I'm working in the lab is I'm always concerned about my safety and the safety of other people I work with. So as we're walking through the lab, if you see anything that looks like safety equipment or things that are in place to help protect the scientists who work in the lab, just note that in the chat too, and we'll see if you happen to see everything. So when we come in the lab, I apologize, this video is a little bit shaky. We have a lot of communal equipment in the lab that everybody uses, including a microscope that's covered there with a blue cover. And there are 12 to 15 different scientists that work in this particular lab. I am one of them. We're approaching my bench. There's my fume hood that I use if I'm working with hazardous chemicals. And there's my bench space where I do all of my reactions. I am one of 3 plant biologists that work in the lab. And there are also a number of organic chemists who are involved in synthesizing different chemical compounds. And we also have some engineers in the lab that are studying different things. A lot of us also do something which is called analytical chemistry. This is really important when we're studying the plant cell walls because these experiments allow us to break down the components of the cell wall and see what's there and see what has changed. And then we can use all of the instruments that are in this instrument room that we're approaching here. And we use any number of these instruments to see how the cell wall has changed and if we succeeded in making changes to the lignin or not. Okay, so what I'm going to do is I'm just going to walk you through what a typical project looks like for me and what kind of things are involved as I'm trying to make changes to the lignin in a plant. The first thing that I have to do is, as I mentioned earlier, I am trying to put products into the lignin that'll make it more easy to break down, or easier to break down. There are enzymes that can make those products that will end up in the lignin. So I need to first identify an enzyme of interest. And once I've identified that enzyme, the first thing that I have to do is I need to test it in the lab, just in a test tube, to see, does that enzyme even do what I think it's going to do? If I feed it this one substrate or this one compound and this other compound, will it make the product I'm expecting? So what I'll do is I will set up a mock reaction, just as I'm here in the vial, where I add one substrate and then I'll add the other substrate to the same vial. And then I'll add an enzyme to the reaction. This is oversimplified. A lot of times there's other components that have to go into the reaction to mimic what the enzyme would see within a plant. But mix it up and then I let it sit for half an hour or an hour to try to make the product. And then the next thing I do is I run it on one of our machines in the instrument machine. This one involves liquid chromatography and mass spectroscopy. Basically what it's doing is it's separating out the molecules based on their, what we call polarity, or their charge. And then we break it into lots of fragments. And by breaking the molecule into lots of fragments, that gives us information on what the original molecule looked like. So if, for example, I tested 3 different enzymes. I had 3 different enzymes that I thought could be really good at modifying the lignin. And each of them produces a product indicated by either this black peak or this pink peak or this blue peak. Which one do you think I would most likely pursue and move forward with for the rest of my experiments? And if you want to answer in the chat, go ahead and do that.
Rebecca, we did also have another question come up during your previous video when you were mixing the enzymes and the products or the reactants. What tool are you using to do this?
When I am setting up, so for when I'm setting up the reactions, do you think that's what it's about? Oh, so I'm using a pipette. That's just a manual pipette. You can also get electric ones, but I'm a little bit old school, so I use a manual pipette, and it comes with tips that are specific to each pipette. For those who said number 3 or blue, you are absolutely right. This is the enzyme that gave me the most product, at least in the test tube. So the odds are pretty good that it's the one that would be most effective in plants. So the next thing that I have to do is I have to work back from that enzyme, which is a protein, to find what is the gene sequence for this. And the reason for this is we can't just go around injecting plants with a protein or with an enzyme and hope it's going to work. It probably won't, because the enzyme might not be in the right place or the right time to be functional in the plant. So we might not get into the right tissue. And also when we want to grow these plants out, if we want to grow these plants outside or in a greenhouse, it's really hard to go through a population of thousands of plants and have to inject an enzyme into every single one of them. It doesn't work. So what we want to do instead is we want to get the plant to make the enzyme for us. For that, I have to go online, and there are a number of databases that have a whole genome, so all the genes in different plants sequenced, and I find the gene sequence for my enzyme. You may have learned in your biology class that genes are transcribed into RNA. So DNA is transcribed into RNA, and then the RNA is translated into a protein, and our enzyme is a protein. So what we're trying to do is put the gene into what's called a bacterial plasmid. A bacterial plasmid is a circular piece of double-stranded DNA. Bacterial plasmids are found all the time in all sorts of bacteria like E. coli or another bacteria we use called Agrobacterium. And what this bacteria enables us to do is that it will cut out a chunk of its DNA, infect the plant, and with its chunk of DNA, the bacterial DNA it puts into the plant, it'll put our DNA in there too, the one that we're interested in that encodes this enzyme. Once that DNA is in the plant, then the plant will make the enzyme for us. And every generation of plants after that will make the enzyme for us. So I design this on the computer and I figure out this is how in the lab I'm going to cut and paste my gene into this bacterial plasmid. And then in the lab, I put it into the bacteria. And as you can see on this plate, every single white dot is a bacterial colony that has this plasmid with my gene in it. Any of these little blue colonies, those ones, it did not work to put the plasmid into the bacteria. It just wasn't successful. So from here on, then I'll just be choosing the colonies that have my plasmid. The next thing that we do is this gets put into plants, which is kind of a long process for some plants. But the plants I put it in are Arabidopsis Thaliana, which is a model plant. And if any of you have worked at all with a plant called fast plants in your classrooms, this is related to Arabidopsis. Arabidopsis was one of the first plants that we knew every single gene that this plant made. And it grows very quickly and it's very easy to grow in the growth chamber, and it's very easy to get bacteria to put their genes or to put our gene of interest into this plant. So it's one of the reasons that we use it as a model plant. The other plant that I work with is sorghum. This is my bioenergy crop plant of interest that I study. Now, in theory, the bacteria has put the gene of interest into our plant. But I have to double check to be absolutely sure. So I need to make sure the gene is there. I probably need to make sure that the gene is active in the plant. And I should probably also check to make sure that the protein, the enzyme that I thought I was making, is also there. So what I do is I collect some plant tissue and then I grind it in liquid nitrogen. And the reason for grinding it in liquid nitrogen is this ensures that all the plant material that all the genetic material and all the proteins remain in exactly the same state that they were when they were on the plant. If you do this at room temperature, what can happen instead is that all the DNA can kind of fall apart. A lot of things can start degrading. Proteins can change their form and then they won't work anymore. And so it doesn't give us an accurate snapshot of what happens in the plant. What the ground plant material looks like is shown here on the right. So it's just ground into a very fine powder at this point. This is the point at which I would do, for example, a DNA extraction. And if you've ever done a DNA extraction from fruit or from some other organism, you know basically most of the steps that are involved in this. We add in a few extra steps to make sure that our DNA is pure in the end, but it's the same sort of process. The problem though, once I've extracted all that DNA, is that I have one copy, maybe two copies of the gene that I'm interested in all of the DNA of the plant. That's not enough to be able to see it. So I use this machine called a thermal cycler and a reaction called polymerase chain reaction so that I can make lots and lots of copies just of the gene I'm interested in. And the next thing I do is I run something called gel electrophoresis, where I add some dye to my DNA sample here, as you can see here, and then I load it into a gel, and then I run voltage through the gel so that the DNA will migrate. The reason this works is that we have a negative electrode up at the top, and then in red at the bottom, which you can't see, we have a positive electrode. Because DNA is negatively charged, it's going to move from the negative electrode towards the positive electrode. And it's also going to migrate based on size. So if you were running DNA in the gel and it's running towards the positive electrode, which size of DNA do you think is going to move the furthest? Do you think larger DNA is going to move the furthest or smaller DNA is going to move the furthest? I'll give you a second if you want to type your answer into the chat. Yeah, I'm seeing smaller from everybody, and you're absolutely right. The smaller DNA is going to migrate the furthest because it's smallest, so we can move through the gel much more quickly. This is an example of what that finished gel might look like, just when I took a picture of it. You can kind of very faintly see the wells up at the top here, and then you can see the dye that we put in with our DNA and how it's migrated over time. When we look at the gel under UV light, this is obviously a very different gel, but it serves as an example. We have what we call a DNA ladder on the left here. Each one of those individual bands represents a different size of DNA. This is how we determine if our product is the right size. Back when I was doing that work on the computer and figuring out what does my gene look like, I also figured out what size is my gene, how many nucleotides is it? And once I know how many nucleotides it is, then I can figure out, is it the right size on this gel? And if it is, great. I probably have my gene in the plants, and then I can continue on to do other analysis on these plants. So my gene is in the plant. I grow the plants until they're fully mature. Then I harvest the tissue and I grind the tissue. This time I just do it at room temperature. It doesn't have to be frozen. I'm really just interested in what's happening to the cell walls at this point, and those aren't going to be damaged at all. And then I can do a number of different chemical analyses at this point. For example, I can find out how much lignin is in my sample. To do this, I'll take a little bit of this ground material. I will dissolve it in a solution of sulfuric acid. That's going to dissolve all of the sugars, and all the sugars will remain in the liquid, and all of the lignin is going to pellet out. So I can filter it through, and all I'm left with after it dries is the lignin. And then by mass, I can determine how much lignin I have there. You may have noticed there's a difference in color here between some of the samples. This is just a normal plant that doesn't have any genes added to it. And this is one where I made a change to one of the genes that's involved in making lignin. The color difference is something that we see sometimes when we make changes to the lignin, which is really interesting. The other thing I can do is I can take some of that ground material again, and I can use different chemicals just to break apart the lignin. And so the lignin will be broken into tiny little pieces now, and I can see what compounds are there, including, is the product of that enzyme present? Did we actually succeed in making it? Is it in the cell wall at all? And I can run it in a different machine, which involves gas chromatography and mass spectroscopy. And this is kind of what the readout looks like. We have a whole bunch of little peaks. And in our lab, because we do a lot of this work, we know what a lot of these different peaks are, based on compounds that some of the organic chemists have synthesized for us and then run in this machine. And I can compare the difference between these peaks between a number of different plant lines and different samples to see, is there anything different? Did we make the product? Is it actually there? How much is there? Is it enough that it's actually going to change the lignin and change the digestibility, or how we can break down the lignin? The other thing that we can do is we can take a small amount of plant sample, we can dissolve it in a solvent, and then we can run it on something called an NMR, or nuclear magnetic resonance spectroscopy. This is kind of like an MRI machine, but instead of imaging parts of your body, we're imaging chemicals that we make, or we're imaging the plant cell wall. And we end up with a readout that's like the spectrum that's on the right side here, where we have different peaks. This is kind of like a topographical map. The bigger the peak is, or the bigger that blob is, the more is present in the cell wall. And so that gives us a lot of information on how the lignin has changed. Again, did we make our product? Is it there? Can we see it? All of these things together lead us to ask the question of, did we manage to engineer a better bioenergy crop? Did we manage to engineer the lignin sufficiently that it's going to make it easier to get to the sugars in the cell wall so we can make fuels? Or did we change the lignin enough that we're going to be able to make specific bioproducts from the lignin as a result? Now, I've talked through this entire process in, I don't know, 10, 15 minutes, maybe longer. The actual time it takes to run this can be years. The reason for that is there's a lot of getting to a certain point and things aren't working, so you'll have to go all the way back to square one again. and find new enzymes and try again. Or you don't see any changes in the cell wall, so you have to go all the way back to the beginning and try again. And we also have to look at many generations of plants, because you need to make sure that that gene is staying in amongst the plant DNA, that it's not being lost and that it's not bouncing around all over the place. We don't want that either. And so we need to make sure it is what we call stable within the plant. So this can take years, but by the time we have an energy crop we're really interested in, then I can hand it off to the other people in the center who are actually making the fuels, who are breaking apart the cell wall components, who are feeding it to bacteria, who are making these products. And they, at the same time that I'm making these crops, they are developing methods that's going to, that they're going to be able to apply to the plants once I provide them with the plants. So I'm just step one in a much larger process into making the fuels. So before I pause and turn it over to questions, oops, sorry. I just wanted to throw this out there and say, come visit us. Right now, we're still operating under pandemic conditions, so we can't do a lot of the tours and the field trips that we typically do, but reach out to us and find out what we do have available. And we have a website here that lists all the field trip and tour options. Everyone in this building is really excited about their research and happy to talk about it with you. So feel free to reach out to us.
What a super cool talk, Rebecca. Yeah. If anyone has any questions or comments, please leave them in the chat or in the Q&A, and I'd be happy to ask them for you.
It's Friday afternoon, everyone's scienced out. So much good science going on.
It's only day 2 of the festival. We got 2 more days. We're halfway there. Isn't that crazy? Someone said.
Yes, it is frustrating sometimes, absolutely. Especially, it can be a little bit repetitive sometimes, but on top of that, thinking that you have the perfect solution and you've got the great idea and this is going to work. And then you can never predict how plants are going to respond and if they're actually going to do what you hope that they will do or not. And when they do, when they don't, you know, that's the plant's mechanism of protecting itself, because it still needs to grow. And that's one of the things we think about a lot. We always try to make our modifications in such a way that it does not affect plant growth or development or productivity. It's not affecting how many seeds the plants make. We want them to be really healthy. We just want to make it a little bit easier to break apart that cell wall.
Yes, I can relate to that. Rebecca, I had my PhD product was basically doing a lot of the techniques that you were using in terms of developing plasmids and then putting them in plants. And it can be a little bit frustrating, but when you get there, it's like, wow, yeah. I did it finally.
Yeah. What advice do I have for people who might want to be botanists or scientists? My advice is reach out to people who are botanists or scientists. You don't have to know them. I have an undergraduate student who is working with me right now, who's been working with me for the last 4 years. And she reached out to me as a high school student saying, you know, I'm interested in biofuels and bioenergy, and I would really like to know what you do in the lab and what it looks like. And so I invited her to come in and we talked for a little while, and then we took a tour of the lab. And when she started up as a freshman at UW-Madison, she reached out to me and said, I would really like to get some lab experience. If there's any chance that I could work in the lab or volunteer in the lab, I'd really love that. And I was happily able to accommodate that and have her come work with me. So yeah, I'd say reach out to people who do that. And if there's one particular field that interests you more than other, then pursue talking to those people. I always find it really interesting to talk to people and find out what they do in their day-to-day and what that actually means.
Yeah, asking people is always a really good start, and people are generally really nice and just want to help. And if they don't know, they'll probably point you towards someone who does know.
Mm-hmm.
And we have 1 minute left. So
I guess I can answer this question really quick about, is it common for undergrads to work in labs? Yes, it is here. Every lab that I know has undergraduate students working for them. So if you are a high school student that's going into university, or you're when you're already in college, if there's a professor that does work that you're interested in, or you just like that professor, enjoy their class, it's always a good idea to talk to them and say, I'm really looking for research opportunities and to be an undergraduate researcher. And if they don't have space in their lab at that moment, a lot of times they'll be able to point you in directions of where you can find other labs.
Yeah, great question. And there are specific programs for undergrads too that allow you to work in labs too. But with that, I just want to thank everyone for coming to this field trip and joining us for this Wisconsin Science Festival field trip. Thank you so much, Rebecca, for this wonderful talk. And a recording of this field trip may be available, and we will follow up with teachers about that. Remember that the Wisconsin Science Festival continues with events hosted from organizations across the state that you can participate in. Just check out wisconsinsciencefest.org for more fun science. See you there, and thank you all so much.